Okay, let's dive into something really quite fascinating today. A technology that's, well, so precise it's been recognized with not one but two Nobel Prizes in physics. We're taking a deep dive into optical tweezers.
That's right. It's truly foundational science. Arthur Ashkin, he developed the core system, originally called it the single beam gradient force trap. That work led to his 2018 Nobel.
2018, relatively recent history then.
Yes. And building on that, Stephen Chu used related techniques, specifically for cooling and trapping neutral atoms. That earned him his Nobel back in 1997, so the impact is just huge.
Definitely. So for you listening, what are optical tweezers? We're not talking about tiny metal pincers here.
No, not at all. It's an instrument that uses a very tightly focused laser beam, usually infrared. And with that beam, you can hold and move microscopic things. Think nanoparticles, tiny droplets, even living cells or bacteria.
And the forces involved are minuscule, right? Piconewtons.
Exactly. Piconewtons. It's hard to grasp how small that is. Maybe a billionth of the weight of a mosquito landing on your arm. But crucially, it's enough force to hold on to, say, a single DNA molecule against all the random jiggling, the Brownian motion, in water.
Okay, but the core question, how does light, which we think of as just, illumination, actually grab something?
Right. It comes down to two key forces that photons exert. The main one, the sort of pulling force, is the gradient force.
Gradient force, okay.
So the particles we trap, they're typically dielectric. They don't conduct electricity, but light's electric field can polarize them. When you focus the laser really tightly, say through a microscope objective lens, you create a steep intensity gradient. The particle is then drawn towards the brightest spot, the center of the beam, where the electric field is strongest.
Okay, that makes sense for the pull. But light has momentum, doesn't it? Shouldn't it also push the particle?
Yes. That's the other force, the scattering force, or radiation pressure. And it does push.
So why doesn't it just shove the particle right out of the trap?
That's the clever part of the design. The scattering force pushes the particle slightly downstream along the beam's direction. But because the beam is so tightly focused, that inward gradient force pulling towards the center is actually stronger. It overcomes the downstream push, effectively locking the particle near the focal point.
Right, a stable trap. And it's not just about holding things still, is it? The sources mentioned measurement capabilities.
Precisely. This is where it gets really powerful. For small movements, tiny displacements near the trap center, the trap behaves remarkably like a simple spring. It follows Hooke's law.
Like a textbook spring system, but made of light.
Exactly. And because it's so sensitive, this light spring lets us measure incredibly small displacements. We're talking sub-nanometer scale. This turns the optical tweezers into an instrument for measuring tiny forces, like those generated by biological motors inside cells.
Wow. And I gather the physics model changes depending on the particle size.
It does. If the particle is much larger than the light's wavelength, we tend to use ray optics. If it's much smaller, like an atom, we use the electric dipole approximation. It ensures accuracy across scales.
Okay, so applications. You mentioned biological motors. What else are these used for?
Well, in biology and medicine, it's incredibly useful for manipulating single cells, grabbing a bacterium, holding a sperm cell, sorting blood cells, even stretching DNA. And as mentioned, really characterizing those molecular motors. But you have to be careful.
Careful how?
Heat. Lasers deposit energy. For live biological samples, you typically use a specific type of infrared laser, like an ND.YAG laser at 1064 nanometers.
Why that specific wavelength?
Because water, which makes up most of a cell, absorbs very little light at that wavelength. It minimizes heating and damage.
Avoids what some call opticution, frying the cell.
A very good term for it, yes. Opticution is definitely something to avoid.
So beyond biology, you mentioned atoms earlier.
Yes, that's a huge area now in quantum science. Optical tweezers are used to trap individual atoms that have been laser-cooled to incredibly low temperatures. Researchers can arrange these trapped atoms into precise patterns, like arrays. Think hundreds of atoms held in place by light.
Like building with atomic Lego blocks. What's the goal there?
Building quantum computers and quantum simulators. In 2021, they demonstrated arrays with 256 atoms, all individually controllable. It's a really promising platform.
Amazing. But creating hundreds of traps, does that mean hundreds of lasers?
Not necessarily. There are techniques like holographic optical traps. They use special optical elements, diffractive optics, to split a single laser beam. This lets you create hundreds of traps simultaneously, all from one source, and you can even arrange them in complex 3D patterns.
Efficient. Is there anything even more, I guess, scalable?
There is. It's called optoelectronic tweezers, or OET. It's a bit different. It uses low-power light, even LEDs shining onto a photoconductive surface. This creates electric field patterns that manipulate the particles, the big advantage, scale and power.
How much scale?
OETs can manipulate something like 10,000 cells or particles at the same time. And they use vastly less power, maybe 100,000th of the power needed for direct laser trapping.
From single atoms to 10,000 cells. Light really is an incredibly versatile tool. It
Right.
really drives home how we can use just the momentum and the gradient of light to act like tiny hands.
Absolutely. And perhaps a final thought to leave you with. We've talked about trapping tiny particles, but the principle, optical levitation, isn't strictly limited to the microscale. Theory suggests, and experiments are exploring, levitating macroscopic objects with light.
Objects you could actually see, held up only by light against gravity.
Potentially, yes, especially if you engineer the object surface with nanostructures to enhance the interaction. The idea that light alone could counteract gravity for visible objects, well, that's quite something to ponder for the future.